Hello friends, I am Gaurav Mukwana. I welcome you all to my another video tutorial of a Biotech guide. So in this video lecture we are going to discuss about a protein characterization. Protein characterization is the part of a downstream processing. As you know, what is downstream processing? So basically it happens after the completion of any bioprocessing or a fermentation process. Where what happened? Basically various techniques or methods are applied for purify and the quantify of a last product which we fermented or which we bioprocessed. Okay, so when we are going to talk about a protein, so for protein various experimental methods that allow for the detection and isolation of the protein is defining as a protein characterization. Okay, and that protein characterization involves this kind of methods for purification and the quantification. So first one is a protein quantification, protein identification, structural studies, protein purity, physiochemical studies, and functional characterization. All combining methods are defining as a protein characterization. So in this video lecture we are going to discuss about these two methods, protein quantification and protein identification. So stay tuned with us and enjoying this video lecture. Before that, let us revise protein structure. As you can see here, protein is made up of more polypeptide chains. Okay, one or more polypeptide chains. So here is a one polypeptide chain. Here small round acid is defining as amino acid and forming a polypeptide bond. And this kind of a chain. Okay, this is defining as a polypeptide chain. One or more chain combine and form a structure is defining as a another secondary level of a structure. Here you can see and form a alpha helix and a beta acids. This two kind of a structure is another having importance. We have already uploaded videos regarding protein structure and function. You may visit in our description box, link given in there. Then after tertiary structure is a nearby the quaternary structure. 3D structure is there basically where this kind of a primary and secondary combine and form a folded type of a complex molecule structure is defined as tertiary. And after completion of this structure, all kind of a residue and the all kind of a formation basically is done as a conformational state. Okay, at the end of a conformational stage, protein is completely in the form is defining as a quaternary structure of a protein. So this is all about of a protein structure. So let us start first with the protein quantification. So in protein quantification, various techniques or methods are used, basically biochemical method. So in this topic, basically I have included 6 major or important method. As you can see here, first one is a UV method, then ninhydrin method, then bisinonic acid BCA method, then Bradford method, trichloroacetic acid precipitation, silver binding procedure. So brief, we are going to discuss in the chart form. Basically I include mode of action, advantages and disadvantages. So stay tuned. Let us start first UV method. UV method is used for a aromatic amino acid compound like as a tryptophan, tyrosine and a phenylalanine, because they use specifically 280 nanometer wavelength of a light, so they can be easily detected via UV method. Advantages: simple, fast, non-destructive, and disadvantages: low sensitivity, identical concentration, and different protein will produce a different absorbance value. So this is first our UV method. And second, acid digestion, ninhydrin method. Here acid and also ninhydrin is used, that's why this method is called as acid digestion or a ninhydrin method. Basically in this method protein is hydrolyzed and form amino acid, and that amino acid is incubated with H2SO4 at 100 degrees centigrade. Now after this incubation a product is formed which react with a ninhydrin, and that product is absorbed 570 nanometer wavelength of light. Okay, so this is the procedure or mode of action. Then advantages: sensitivity is there, phenotypic and aromatic compounds do not interfere with this assay. And disadvantages, hazardous chemical is used like H2SO4, so that might be called a non-destructive. Then various methods, another bioassays acid BCA method is used for the basically copper containing reagent is there. Okay, in this reagent, BCA reagent, copper containing reagent is there, which is reduced via protein. Then after that reduced reagent react with a bioassays acid, and a derivative is formed which absorb 560 nanometer wavelength of a light. Advantage is sensitive, less susceptible. Okay, so these are the advantages. And for disadvantages, cost, then departure extract assay protocol, and those are the another temperature concentration. Those are the another reason for non-applicability of this method. Then Bradford method, as we know, pH dependent method where commercial brilliant blue dye G250 is used for the protein absorbance, and maximum 595 nanometer wavelength of a light is detected via this method. Moderately sensitive, easy and fast to run. This is the advantages. And alkaline pH buffer will not interfere with the assay, subject to interference by detergents like sodium rhodiolysyl sulfate. So these are the disadvantages of this method. Then another method, trichloroacetic acid precipitation method. Basically this happens in the addition of a TCA, this chemical trichloroacetic acid, to a sample in order to precipitate it, and followed by resuspension of the protein and assay by one of the above method. Okay, so assay basically happen by one of the above method, the spectrophotometer. And basically in this method what they do, addition of a TCA, and after precipitation is applied, and after that resuspension is applied. Okay, and assay will be done by spectrophotometry method. Advantages, used to remove non-TCA precipitate interfering agents from the protein sample before quantification. So this is the advantages. And disadvantages, laborious method is there. At last, a silver binding procedure. So based on the binding of a silver ions to protein, this is the mode of action. Advantages, extremely sensitive. And a disadvantage is chelating agent, detergent and reducing agents interfere with this assay. So these are the protein quantification method. Now we're going to discuss about our second in protein characterization, that is a protein identification. Okay, so as we discussed in protein quantification, different various methods are used for a protein quantification. Similar in protein identification, another three techniques are used for identification. First one is a immunoassay, second is a Western blotting, and third one is a bioassay. So briefly we're going to discuss next. So what is immunoassay? Immunoassay is a immunological reaction that capitalizes binding between an antibody and antigen. Okay, specific antibody is bind with a specific antigen. In this method I include five immunoassay method. First one is a radioimmunoassay. As name suggests, radioisotopes is used. Okay, here radioisotopes is covered with antigen. So why we use radioisotopes? Because our interest, specific antigen is bind with a specific antibody. That's why we labeled with a antigen with radioisotopes. Now this radioisotopes labeled antigen is bind with a specific interest of a complementary antibody. Okay. An example, radio immunoassay is used specifically for identification or hormonal detection. Basically, like here I-131 or I-125 is used for the detection of a tyrosine amino acid residue. So this is the first our radio immunoassay technique. Now second method in immunoassay, that is a counting immunoassay. As name suggests, counting. So here one cell counter application is there. Let us this brief. In counting immunoassay, polystyrene beads is used, which is coated with many antibody which is complementary to target antigen. Okay, so this is the simple principle behind this counting immunoassay. Now this beads and target antigen is incubated with some period. During incubation, some beads remain unbound and some remaining another bound with a specific antigen. Okay, so now this remaining unbound antibodies is counted via cell counter application. So the entire solution is passed through a cell counter where the unbound beads is counted, which is proportional to the amount of a total antigen. Okay, so this is the simplest mechanism behind this counting immunoassay, and for application is used for the hepatitis B antigen identification, then anti-hepatitis C virus identification, also used for anti-TP, that means the Treponema pallidum antibody identification or detection, and also used for the whole blood sample count. Okay, so these are the application of counting immunoassay. Now in immunoassay, third method is there, enzyme immunosorbent assay or enzyme linked immunosorbent assay. Mechanism also, antigen antibody mechanism is also there. After incubation, unbound antibody is washed. Now bound antibody is linked with the enzyme. After that, when this antibody linked with the target antigen, substrate is added, and that substrate react with the enzyme which is labeled or bound with the antibody. So what happen? Enzyme substrate reaction is happen, and that produce a quantifiable color change which is measured. Application, Magento ELISA is used for the CD4 plus cells count in AIDS, also used for the HIV detection and pregnancy diagnosis. Now fourth in immunoassay, that is a fluoroimmunoassay. As name suggests, fluorescent probe is used for the detection. So here antibody is labeled with a fluorescent probe. After incubation, antigen antibody complex is isolated and the fluorescent intensity is measured. So this is a mode of action of a fluorescent immunoassay. Now application. So this technique is useful for the endocrino pathways basically. So first is a CAH is defining as a congenital hypothyroidism, used for the thyroidism detection, also used for the HIV, cystic fibrosis, and congenital toxoplasmosis disease identification. So these are the application of a fluoroimmunoassay. Next, chemiluminescence immunoassay, defining as a CLIA. So this is the similar principle or mode of action is there, as we discussed in a enzyme linked immunosorbent assay or a fluorescent immunoassay here, but reporter is different. What? So let's see. Luminescence is released of a light due to electron. Okay, what happened here? When energy is absorbed, electron being kicked up and goes into higher energy state. After that, in relaxation state, that absorbed energy is emitted as a photon. Okay, and that is detected via this chemiluminescence immunoassay method. Application, luminal. So this is the method where reagent is combined with this kind of a luminal chemical and form a luminescence reaction. Then peroxyoxalate, which is also used for the detection of a less amount of hydrogen peroxide. Then electrochemical immunoassay and also bioluminescence. Those are the application of a chemiluminescence immunoassay method. So here we completed immunoassay method in protein identification. Then second in protein identification, that is Western blotting. Western blotting is specifically used for the protein isolation. So various steps are there for Western blotting. First is sample preparation. So sample is prepared via centrifugation technique applied to a cells. After lysine buffer is added into the protein, at the end supernatant is collected, and now further they proceed for the electrophoresis technique. Okay, see, this is the similar like gel electrophoresis technique, but here another application of is there, that is a PAGE is defining as a polyacrylamide gel electrophoresis. Okay, and here another salt is used, sodium dodecyl sulfate. Now after sample preparation, this electrophoresis is applied For that, two kinds of gel is prepared: resolving gel and a stacking gel. If our protein is small in diameter or Dalton, okay, so resolving gel is used, and if big, so this kind of stacking gel is used. Okay, resolving gel having a higher concentration acrylamide, and here a lower concentration of acrylamide is there, small pore, big pore, and the pH is whereas 8.8 and 6.8. Another concentration is the same, 1.5 molar trees SDS, DH2O, APS, ammonium persulfate, and a TEMED, that is a tetramethyl ethane 1,2-diamine. Okay, so this is the SDH phase preparation. After gel preparation, prepared gel is transferred to glass plate. Okay, and after that comb is applied. Then this gel is goes for the solidifying. After solidified, gel is transferred into the denaturation buffer that having a trisulfide and the pH is 6.8. Then sodium lauryl sulfate, ethanol, bromophenol blue, glycerol, and a DDH2O. Okay. After that, SDS phase electrophoresis is applied. For that, basically vertical system is there, where protein samples and marker loaded in a vertical SDS phase system. Here the buffer is applied. Okay. Here the buffer is there, and here well is created via code. Okay, and here the protein sample is put it down here. Direction of a migration sample is in a vertical SDS phase system, as you can see here, and then after this electrophoresis, is gel goes for the Commissaire blue stain. Next step is a very interesting step, membrane transfer. Now membrane transfer. So complete gel holder cassette is transferred into the tank, which is filled with a buffer, and this kind of accessory is maintained. At the below, sponge is there, then filter, gel which we have prepared, and electrophoresis is done. That gel after this gel above gluting membrane is there, and the membrane is PVDF, polyvinyl difluoride, is specifically used for the protein transfer. Okay, then after filter is there, and at the above weight is there. Now what happened? Trapped protein in the gel is transferred to this bloating membrane. After membrane transfer, membrane blocking application is applied, where first BSA or non-fat dry milk is applied to a membrane, which is bound with a non-protein part. Okay, so like this is the membrane. Okay, and here this kind of a dry milk or BSA is bound with this kind of a surface, which is where the none of any target protein is there. Okay, basically at the background this BSA protein is bind. Now primary antibody is applied, which bind with a target protein. Okay, so here this kind of a target protein is there, this kind of a target protein is there, where primary antibody is bind. Okay, and that is inoculated and washing out. After that, secondary antibody is applied, which bind with a primary antibody. Okay, so this is the primary antibody. After secondary antibody is applied, which is a fluorescently labeled. Okay, fluorescently labeled, which bind with this primary antibody. So after that, at the end, washing is applied, and at the end we have a particular specified protein, specified target protein is there. So by using this Western blotting technique, we get a purified protein or target protein, which we have to inoculate from the sample. Okay, so this is the application of a Western blotting. Third, in protein identification, bioassay technique is there, which is used for the determination of a relative strength of a substance, like as a drug, by comparing its effect on a test organism. Okay, so basically this is used for the identifying the effect of a test organism to a relative substance. Is used for the measure the biological activity, also used for the biological activity and effect of a substance, like as a hormone or a drug on a living organism, as we discussed. Okay, so bioassay is used for that. And for that bioassay, we will going to discuss about a different 1, 2, 3, 4, 5, 6. Okay, so 6 protein we are will going to discuss and their bioassay description. Okay, so let us discuss. So first, interleukin 2 protein having ability to promote the proliferation of a activated T lymphocyte. Then second, interferon alpha having ability to inhibit the cytopathic effect of a certain viruses. Example is vesicular stomatitis virus on a certain human cell line. Then granulocyte colony stimulating factor having ability to promote a proliferation of a certain animal cell lines. Then tumor necrosis factor having ability to induce a cytotoxic effect on a certain animal cell lines. Example is a murine fibroblast cell line. Then erythropoietin having ability to stimulate the proliferation of a TF1 in animal cell line. Then polypeptide antibodies having ability to prevent the growth of a various indicator microorganism. So this is our third bioassay in protein identification. So here we have done two topic of a protein characterization. First one is protein quantification, where we have discussed 6 spectrophotometrically or a biochemical method for a protein quantification. Then second we have discussed protein identification, where we have discussed another 3 sub techniques: immunoassay, Western blotting, and bioassay. In immunoassay we have discussed radio immunoassay, counting immunoassay, ELISA, fluoroimmunoassay, and then chemiluminescence immunoassay. Then second, Western blotting, we have completely understand the procedure of a SDS page, gel electrophoresis, and a membrane transfer. Then after third bioassay, where we have discussed 6 different protein and their bioassay description. So for today's topic, we have done these two: protein quantification and protein identification. Next, we are going to discuss about a structural studies and a protein purity of a protein characterization. So here we completed our topic.